Greetings, classy. Thank you so much for connecting once again. This is end of term. This is end of academic year for amended senior certificate. We are writing exams just now. This paper that I'm bringing to you right now is an exam prediction and scope are based on technical previous papers. So paper two, basically students understand that we have to understand DNA and RNA. There's no way you should go to exam not knowing the structures, the differences, uh, the formation of RNA and all the building blocks of both nucleotides, protein synthesis and transcription. We talk about types of dominance. I guess you remember the complete, incomplete, and codominance. We talk about crosses, that is monohybrid and dihybrid crosses, calculating percentage chance factors, of course, understanding the number of gametes that can be formed or that can be produced in different contexts, whether like dihybrid crosses, of course, sex-linked alleles and diseases. We talk about homophilia, we talk about color blindness, which you do based on, of course, based on family tree diagrams that also comes in in that reference. Evolution is no way that you can go to exam and you don't get it there. So we need to understand evolution, the phylogenetic tree diagram, theories of evolution, evidence of evolution out of Africa hypothesis. We talk about also uh, types of speciation, mostly the geographic speciation. How does it occur? Of course, not forgetting natural and artificial selection. So without wasting any time, I'm saying this paper is actually an exam prediction. It's actually an exam uh, revision template that we are looking at. So it's paper two, as you can see. Without wasting any time, let's just go to what you should expect or what is in the paper. Instructions always are there. I'm not gonna go through them, but please note that every time when you go to exam, instructions are supposed to be read. And then we have got question A or section A, which is multiple choice. You are given various options and you need to give the best option that answers the question. So scientists who discovered Carabo Australopithecus sediba. So now I'm sure I remember the different the various scientists who discovered different fossils in Africa. So you need to know all these and should be able to know what is it that they discovered and where was it discovered. So this one, the fossil Australopithecus sediba, which is Karabo, was discovered by Lee Berger. It was discovered by Lee Berger. I guess you remember very well that we have, of course, a multiple of them which have got their different contributions in terms of African uh, discoveries or also the cradle of mankind in Africa. So which one of the following is a source of variation that occurs during normal meiosis? A source of variation. We are saying a source of variation during normal meiosis. If you talk about normal meiosis, it must give us to say it has to have the number of chromosomes in a cell. Number of chromosomes. It have to have number of chromosomes that are in a cell that are supposed to be formed in a number of chromosomes that are that is, for example, we have got MM, which must say if it is normal meiosis. If we are talking about meiosis, normal meiosis, MM should be equals to, after meiosis, it must be equals to M. We have got 46. After meiosis, it must give us 23. So we have got AB as a gamete producing cell. After meiosis, we must have A, we must have B. So what I'm basically saying is normal meiosis, must always result in the separation or in the halving of the number of chromosomes that are found or that are to be in a gamete. So now, in terms of this, which one of the following is a source of variation that that occurs during normal meiosis? So normal meiosis, it must be is a source. It must be random arrangement of chromosomes, which occurs during meiosis. It results in halving in variation, whereby the gametes that are formed are always different. They don't look the same. They are totally different. Chromosomal mutation is abnormal. That is, of course, when certain changes have occurred, which resulted in the change of DNA nucleotides, which, of course, at the end of the day would cause uh, a different form of protein to be formed. And random mating, of course, it can actually, one of the processes that occurs, but in terms of gametes formation, they are formed during or on random arrangement of chromosomes. Cloning is artificial. Cloning is artificial process of producing organisms that are identical. So it's not in that reference. So how many sex chromosomes does a normal human how many sex chromosomes does a normal uh, human female inherit from her mother? A normal human female, we are saying that the number of sex chromosomes. And one thing again, remember we are revising in this case, in female we say sex chromosomes are XX, and then in male we say that it's XY, which of course I'm saying these are called sex chromosomes. So how many do a normal female inherit from a mother? From the mother, a normal female can inherit, we are saying it can, a normal female can inherit uh, an X from the mother. A normal female can inherit one sex chromosome, which when combined with the male, we can therefore have an XX, or we can have an XY, which I'm saying from each of the parents, a child inherits one sex chromosome, one X chromosome comes. So it is an X plus an X, which results in that, or it can be an X plus a Y, which results in the other process or in the other combination that you are seeing. So it must be known that a single chromosome, sex chromosome, is inherited, which is a and then during which phase of meiosis does the nuclear membrane disappear? The nuclear membrane, it actually disappear during prophase. In terms of meiosis, I say that you should not go to exam also not understanding the process or the phases of meiosis. They might be given in haphazard way, therefore you are asked to arrange them, or you are supposed to give the characteristics that takes place at each stage. Nuclear membrane disappears during prophase, and then which one of the following is an example of continuous variation in human? Continuous variation is an example of discontinuous. That is one other thing which happens here, to say you need to be able to read and identify the question very clear. It is saying discontinuous variation, which means it must be the one with with a limited range of values. That is what you call discontinuous. So in discontinuous variation, we have got, in this case, we have got we have got gender, which gives us male and female. So that when we draw a graph, we can say the number of male children in class of Life Science 2025, there are 40 girls, and then we also have, there are 10 boys. So that when we look at this graph, it identifies that this is a discontinuous variation which can also be seen or can be demonstrated by having a graph that can run and drop. So what happens is that this is an example of a discontinuous variation, which is totally opposite to a continuous. When we look to a continuous variation, when we look at a continuous variation, I think you know that it has got a number of spread. It has got a spread that can also run and drop. That is a continuous variation. So please, you need to be able to understand this, because in many cases, you should know that we have got a range of values which we can say that it is starting from maybe from 2, and then it is ending to maybe something like we are saying the spread, it ranges from there up to the last, which can actually be a broad range of values that are there, which you can see that otherwise we have got that spread, which is ranging is across a number of intermediate values that are involved. So gender is only, we have got gender, we have male and female, which can of course be a different situation. But weight, if you look at weight, weight can be so varied. So what I want you to understand is that gender is what is an example of discontinuous variation, which makes the answer to be C. For a particular characteristic, the offspring inherits dash. A particular characteristic, the offspring, which is, we are saying, the baby being produced, it inherits, the baby inherits one allele from the mother and one allele from the father, which of course you can see that in any case, we have got one allele from the mother and one allele from the father, which we can say, for example, still looking at gender as an example, we are saying one allele which is from the mother, and then from the father, we have one allele as well. We have got one allele from the mother and one allele from the father, which when we combine this, we have got XY, we have got an XY, which you can see that the XY is actually showing that we have got one from the mother, we are saying one from the mother and one from the father, which together they give us that. Of course, we can give it even in terms of height. We can say it is total, which we can say this can be from the mother, that is tall, and that is short. So we are saying the parents are having these characteristics, so it can come from the father, it from the mother, and then it comes also from the what, from the father. So together we always have a situation, a combination of alleles that define a single characteristic. So which the characteristic in this case is what? Is height. So height is controlled by two alleles. That is, we talk about homologous and homologous and homologous chromosomes. At the same time, homozygous and heterozygous combination of alleles, which I want you to take note of. So the answer is what is A. And then which of the following is correct for speciation through geographic isolation? I said that types of speciation, we have got geographic, and we've got also reproductive isolation, which also is another form of speciation, reproductive isolation or geographic. Geographic, we need to understand that the population is separated by a physical barrier under geographic. So which of the following is correct for geographic speciation? Is correct. The question is saying is correct. So what is correct with reference to geographic speciation? Each population undergoes natural selection independently. So the independent undergoing of natural selection results in two different population species, two different populations developing. So each population undergoes natural selection independent from each other. So in that case, we have got population A that actually separated into two groups. the two groups, we have got population B and we have got population C. So now here we have got a barrier that separates the two population species or the two population together from each other. So now what would happen is at the end of the day, we are having B as a separate population and also C as as a separate population. It is because of independent natural selection, which results in genotypical, I mean, each population being genotypically and phenotypically different from each other. So that is why you see that. And then uh, below is a list of events that occurs during cell division. List of events that occurs during cell division. So we've got homologous chromosomes line up at the equator of the cell. Chromatids are put to opposite poles. Chromosomes pair arrange themselves randomly at the equator of the cell. Individual chromosomes line up at the equator of the cell. And then which of the following combination occur in both meiosis and mitosis? I said again that meiosis, we can say meiosis 1, and then mitosis, we can also define that as what is meiosis. This can also be meiosis, meiosis 2. Meiosis 2 and mitosis is one and the same. That is one thing that I want you to take note of. So is a combination of both meiosis and mitosis. So in both mitosis and meiosis, we have got number we have got in both mitosis and meiosis, chromatids are put opposite poles of the cell. It occurs during mitosis and meiosis, and then individual chromosomes line up at the equator, which also is correct to say that individual chromosomes line up at the equator. It also correct. So what happens is that it makes D the right answer to say that the lining of chromatids are put to opposite poles of the cell and individual chromosomes line up at the equator. They occur in both meiosis and mitosis. And then a short piece of DNA containing 19 nucleotides in each strand was analyzed. The number of chromosomes of the different nitrogenous bases in each strand is shown below. So one thing which I want you to understand is that when we look at DNA strand, when we look at a DNA strand, this I have already calculated it before again. It's just a repetition of it's a repetition of questions, which I want you to always note that what happens is that the number of nitrogenous bases of complementary bases is always the same. So what happens is that A is equals to T, which means that D, C is equals to what? Is equals to G. So in other ways, we are saying A, the equation that can be formed here is that A times 2 plus C times 2 is equals to total number of nitrogen bases is equals to total number of nitrogen bases in DNA molecule. So now, what you need to understand is that if we have got A, if A is 7, it means RT also is equals to what? Is equals to 7 as well. That's what happens there. So I'm saying we once calculated this question as well previously, saying that how many nucleotides containing thymine were present in the DNA, in how many nucleotides containing thymine were present in strand 1. In strand 1, how many nucleotides containing thymine were present in strand 1? That can also be seen that I say that if you look at that, it is worked based on number of nitrogenous bases that are there. So if you look at this, I'm saying which have got thymine bases that are present in that regard. It can also give you that, that will give you to say that is equals to, it's gonna be also equals to, it's gonna also be equals to 4. It's gonna also be equals to 4. What I'm saying is the formula is calculated as I shown, that A times 2 plus C times 2 plus C times 2 is equals to total number of nitrogenous bases that are in a what, in a DNA structure. So strand 1 and strand 2, which we are also given there. So now what we only supposed to understand is that the number of nitrogenous bases that are in strand 1 and strand 2, the complementary pairing fact remains the answer. Complementary pairing of bases is the formula that we always use in that respect, which is equals to what I have shown you, that A is equals to T to C. I mean, A is equals to T, C is equals to G. A times 2 plus C times 2 is equals to total number of nitrogenous bases that are in there. And then we give the biological terms in the following statements. The biological terms that are defining the following statements are the process of change in the characteristics of biological species over time. The process of change in characteristics of biological species over time. This is also known as evolution. It is evolution. It is called evolution. So you know, evolution is a slow gradual process through which a species evolve or change over time, which can be based or which can be otherwise resulting in development of new species, taking it from the concept that mankind evolved from the primate Homo species. So the change of characteristics of biological species, living species over time is evolution. The type of bonds between nitrogenous bases in DNA, I'm sure that you remember and surely know that uh, these are called weak hydrogen bonds. I want it to be so precise. Weak hydrogen bonds. They are there to say that we have got A, we have got T, and then in between there, we say this is called a hydrogen weak hydrogen bond, which joins the complementary bases together. And then the structure that join two chromatides of a chromosome, two chromatides of a chromosome are joined by a centromere. It's a centromere. We have got that piece. That is a chromatide. That is a chromatide. This is what you call a centromere. The piece that is joining the two pieces, the one that joins the two pieces together is what we call the centromere. That is a chromatide. That is a chromatide. And therefore, this is a centromere. The centromere join the two pieces together. Division of cytoplasm of a cell during cell division is called cytokinesis. That is called cytokinesis. Cytokinesis, the division of cytoplasm of cell during the cytoplasm of cell during cell division, and then the process during meiosis where there is an exchange of genetic material between chromatides that is called crossing over. I'm sure you still remember that crossing over is also variation in meiosis. And then the structures in animal cell that give rise to spindle fibers during cell division. The spindle fibers, the structure that give rise to them is also known as, we say that those are called centrioles. Centrioles. the similar structures that are inherited from a common ancestor and are modified for different functions, which we say that evolution that shows that organisms are evolved from the same or common ancestor, we say they are called homologous structures. Homo means the same, homologous, homologous structures. And then the phase in the cell cycle during which DNA replication takes place. DNA replication, it occurs during the interphase. DNA replication occurs during interphase. Organelle where translation occurs during protein synthesis. The organelle where translation, where do we have translation occurring? It occurs it occurs by the ribosomes. The ribosome is which reads. I'm sure you remember last time also check on metric hub, which we say that is mRNA, which also have the ribosome there. And then this one has to pass through the ribosome, reading 3 by 3. And then the order of the codons versus the anticodons that comes from the tRNA comes together, and then that read, the ribosome actually is where translation occurs. It identifies the order of codons, picking up the corresponding anticodons from the tRNA. Look again from, check again from metric hub. There is quite enough explanation with reference to this. We got question 1.3. The type of evolution characterized by long periods of little or no change, alternating with short periods of rapid change. I'm sure you remember that when we are looking at this evolution, which is characterized by long periods of little or no change, alternating with short periods of rapid change. You remember the other theories of evolution, punctuated equilibrium, versus gradualism. So punctuated equilibrium, long periods of little or no change, and then alternating short periods of rapid change, which makes B only to be a right answer. Artificial selection, nothing to do with evolution, but it's just a human force in terms of the selection of nature. Plant with white flowers that is crossed with a plant with red flowers and produces offspring with pink flowers, which we say that if you look at this, it is called incomplete dominance. It's called incomplete dominance or partial dominance, which makes A to be the answer. A only. We said red plus white, it gives us pink, which of course is called incomplete dominance. Complete dominance, this one we have it when TRO is crossed with what? When TRO is crossed with short, and then in that case, this results in what? This is equals to TRO. Where TRO, you can see that TRO is the dominant allele and the recessive allele is taken over, or it is overpowered. The separation of alleles during gamete formation, it is actually a principle of random segregation. A principle of random segregation, be only. Moyamoya is a disorder caused by dominant allele R. This disorder damages the arteries supplying blood to the brain. Pedigree below shows the inheritance of moyamoya in a family. So please take note that the disease is caused by a dominant. It is caused by a dominant allele. That is one thing. And please always mind that, because quite oftentimes we know that it is caused by recessive, but in this context, they are saying that it is caused by a dominant R. So now, always you should know that I was always trying to ensure that you guys, when you go to exam, you know that we have got various possibilities that arise here to say we have got affected and unaffected, and when we have got careers. So when you go to exam, note that we can say female here, I can say female, and then I can say male on the other side. So for male, we have got XY, which we say that that is close to affected male. We have got XY with the small with a small h, with a small r, with a small r is equals to normal, is equals to normal male. And then for female, we have got XX, of course, I mean, R, of course, with R, and R is equals to affected, is equals to affected, uh, R is equals to affected female. We have got X, and then we have got R is equals to normal, unaffected. We have got R is equals to career, female. So what I'm saying is, whenever you go to exam, take note of these representations so that you would be able to calculate them. And one funny thing about it is that a career can also be given by an unshaded circle, unshaded shape. Unshaded square can also be representing the female who is unshaded, can also represent a female who is what? It can also represent a female who is a carrier. So you shall see that this can also be represented by a clear circle, but there is some key areas of concern that I'm going to demonstrate here. So now, what is happening is how many generations are represented in the diagram. The generations, we said there are a number of, we are looking at the profiles that we have, which are representing people who have lived at a certain space of time. We say that is a generation. So we are saying we have got, this is one, it's a generation. We have two, and then we have got also three. The profiles that we have are the ones that we use to calculate a generation. So A and B are a generation. C, D, E, F are a generation. H, I, J are a generation. So there are three generations, as you can see from there. So how many generations are represented there? This is equals to, there are three of them. Give the letters of an affected male. The letter or letters of an affected male, unaffected male, unaffected male. As you can see, we said that male are represented by a square. Male is represented by a square that is unshaded. So unaffected male, as you can see from the graph, we only have H. H is representing unaffected male. Is the only one that we have, which makes the answer to be what? Which makes the answer to be H. H only is the one that is unaffected in that reference. Genotype of individual A. Genotype, we said, is the one that is the genetic makeup given by letters. Genotype of individual A. Individual A is actually, we have got individual A is a man who is affected. So an affected man, this man is affected So as you can see there, we are saying that it should be man is given as I've given you there. So this man is gonna be given by X, Y, and then the man is affected, is an affected man. So it is X, R, Y, that is individual A. And then letters of individual not biologically related to A and B. Not biologically related, it means they come out of the family line. Not biologically related from A and B. Not biologically related to A and B. These people must come from outside the family of A and B. So these can be people who are marrying into their families. These are people who are marrying into their families. So as you can see here, we are saying C is a son-in-law who married this daughter, and also F is also a daughter-in-law. So we have got C, we have got F. They are marrying into the family. They are not descendants of A and B. These are not descendants of A and B. So it is C and F. C and F. C and F are not biologically related to A and B. C and F are not biologically related. And then question 5 represents part of an abnormal human karyotype. Karyotype, we say this is the structure, is the arrangement, shape, and number of chromosomes that are in the human cell. The structure, the arrangement, the shape, and number of chromosomes that are within a given human cell. So the question says, it says how many autosomes are shown in the diagram. How many autosomes are shown in the diagram? Autosomes, remember we said that autosomes, there are any other chromosomes other than 6 chromosomes. These are chromosomes other than 6 chromosomes is what we call autosomes. So as you can see, we are counting 21 and 22, which gives us 3 plus 2, which is equals to 5. We have got 3 plus 2, which is equals to, we have got 5 there. And then name the type of chromosome represented, the type of chromosome represented by 23. 23, I told you before again from channel that this represents what are called 6 chromosomes. It represents 6 chromosomes. It represents 6. It represents 6 chromosomes, which we also say that they are called gonosomes. Gonosomes or 6 chromosomes is one and the same thing. 23 represents the 6 chromosomes. The question now comes and says, next question, name the disorder represented in the diagram. The disorder that is represented, we have got trisomy 21 here. 21, we have got, it is giving us a trisomy 21. So trisomy 21 is caused by non-disjunction of chromosomes. This is called non-separation of chromosomes. non-separation of chromosome 21, which sometimes they call it is also called non-disjunction. Is one and the same thing. Non-disjunction, non-separation, non-disjunction or non-separation is the disorder. And then process during anaphase of meiosis that resulted in the abnormal number of chromosomes in this karyotype. The process during anaphase, which okay, in this case, the question is saying the disorder first, the disorder is actually trisomy 21, and then it is caused by, it is trisomy 21, the disorder is trisomy 21, which is also called Down syndrome. It is called Down syndrome, which process of anaphase we are saying that is caused by this, it is caused by non-disjunction or non-separation of chromosome 21. Non-disjunction or non-separation of chromosome 21. And then state the gender of the represented karyotype. The gender, I said that the gender is defined by chromosome 23. This 23, it defines the sex chromosome, as you have seen there to say it is known as the sex chromosome. So this long is equals to X. The short is equals to Y. So this together is equals to XY, which is equals to it is equals to a male or a boy, male individual. It is equal to a male individual. If you look at this, joined together, the result in giving us an XY, which is a male. The short, it's called short Y chromosome. It represents a short is a Y and the long is the X, which is giving us XY. Hence, it is the what? This is a male individual. That's a male individual. We are given in rabbits, brown fur is dominant to white fur and long ears is dominant to short ears. So what we are given here is that we are given fur color. We are given fur color. Fur color is what? Is actually we have got brown is dominant to white. We have got brown also, which is represented by B, and also white, which is represented by small b. And then we have got ear length. Ear length is equals to ear length. We have got long ears. We have got long, which is represented by E, and also short, which is represented by small e. So as you can see there, the question is saying a rabbit that is heterozygous for both characteristics. A rabbit is heterozygous for both characteristics is crossed with a rabbit is crossed with a rabbit with short ears. A rabbit that is heterozygous for both characteristics is crossed with a rabbit with short ears. So heterozygous, remember to say heterozygous is a combination of a dominant and a recessive. So it is it is BB, or we also have, we are saying, and also it is also white rabbit. We are saying is crossed with a white rabbit, is crossed with a white rabbit with short ears. So this one is that we are given this as to say that is BBE is crossed with white, which is white is small b and short ears, that is what is happening. They are crossed like that. So name the type of cross that is shown. Give this is called a hybrid cross. Is called a hybrid cross, which we are saying two characteristics are considered or are involved at a single time or at a single cross. Is called dihybrid cross. Give the phenotype of a rabbit that is dominant for both. Give the phenotype of a rabbit that is dominant for both characteristics. Phenotype, we said, is the outward appearance. So that is dominant for both characteristics. So it is BBEE. That is the genotype. This is the genotype. So in terms of its phenotype, it's going to be brown. It's actually going to be brown and long ears. Is brown and brown color. You can say brown color and long ears is the what? This is the phenotype. The phenotype we said is the outward appearance. Is the outward physical appearance of an organism that is defined, outward appearance, which is defined by what? By the genotype. The genotype gives us the phenotype. So our answer, the phenotype is brown and long ears. That is our answer. Our answer is brown that is the phenotype that we are talking about. Brown color and long ears is the phenotype that we are talking about. Is the phenotype that we are referring to. And then genotype of the white rabbit with short ears, give the genotype of a white rabbit with short ears. It is already given also here, saying that is crossed with a white rabbit, is crossed with a white rabbit, is crossed, we are saying it is, we are saying white rabbit and short ears. White rabbit is this one, which I have given already in my question here to say white is represented by small b. White is represented by small b. Short ears is represented by small e. So that is to say that if we say the genotype is given by small letters, white is small b and short ears is small e. That is the genotype that you are supposed to give. Genotype of the gametes of a genotype of the gametes of a heterozygous brown rabbit with short ears. So genotype of the gametes. So what we are saying is that of a brown rabbit with short ears. So brown rabbit with short ears, we said that brown, which is heterozygous for brown eyes with short ears, it is like that. Brown rabbit with short ears. So now we are saying the gametes that are going to be formed here. This is a gamete producing cell. So now when that is producing gametes, we must just have a combination of B and E and small b and E. So we are going to have this one. We are going to have small b, it goes with the small e, and then it gives us a b. I mean, it's a big, it gives us a dominant b and small e. We also have small b, it goes with that small e. It gives us a be. So these are our gametes that we are talking about. We have got two gametes that are going to be formed, which is cause to say that we have got these two are the gametes that are produced. The gametes produced, which is cause to Be and also Be. So that is our answer there. That is our answer. I also have refer also to metric hub for more questions on gamete producing cells, on ways of calculating number of gametes produced. Refer also to metric hub on the question on number of gametes that can be produced there. We have got a nice video which has been calculated for you to work on this. We are given a diagram that represents transcription and protein synthesis. This also, guys, has been done severally on metric hub. This has been done severally on metric hub class. It was done severally. Number of several times that we have done that. For the interest of time, I'm not going to explain a lot on transcription and translation because I have got questions and notes and lectures that are done explaining this. Name the type of cell where the process occurs. Name the part of cell where the process occurs. The process occurs, obviously, like we are saying, transcription, protein synthesis. Transcription, it occurs in the nucleus. Transcription, it occurs in the nucleus. And then protein synthesis, it occurs in the cytoplasm. It occurs in the nucleus and also in the cytoplasm. So identify sugar X. Identify sugar X. So as you can see, this is a DNA structure. So sugar X in DNA is equals to deoxyribose sugar. It's called deoxyribose. Nitrogenous base Y. Nitrogenous base Y because Y is opposite. A is equals to T. So A pairs with T. Okay, this is, of course, as you can see, this you can see that this is mRNA, which is meaning to say is an RNA structure. So A pairs with what? A pairs with U. A pairs with U because this is what we are saying. RNA does not have T. Instead, it has what? It has U. So it is uracil. As you can see that we have got what? Uracil because it's coming from RNA. Tabulate two difference between transcription and differences between transcription and DNA replication. When we are saying tabulate, we are saying draw a table DNA replication, and then we have got transcription. Give the differences between the two, DNA replication and also transcription. So we can say in terms of the differences in DNA replication, the whole DNA molecule is used. One whole DNA molecule is used, and then obviously in this case only a small section of DNA is used in transcription. A small section of DNA is used. And then another difference during the process DNA replication, it results in two identical DNA molecules are formed. Two identical DNA molecules are formed, and then in this case of transcription, mRNA is formed. mRNA is formed during transcription, but during DNA replication, two identical DNA molecules are formed. I'm going to give the last one, which I can say that in DNA replication, both strands act as templates. Both strands act as templates during DNA replication, and then in this other one, transcription, only one strand acts as a template. Only one strand acting as a template. The difference is that you can give with reference to DNA, and the difference that you can give there. I think maybe if I were to add another one, last one, 4, I could also say that DNA unwinds and unzips completely. DNA, it unwinds and unzip completely. However, in transcription, DNA unwinds and zips. DNA unwinds and unzips partially. only a small portion of DNA, it unwinds and unzips. These are the basic differences that I can give between the processes of transcription and translation that you must also take note of the basic differences. So when you are saying tabulate, draw a table. That's what it means by tabulating. And then we have got a mutation occurred on a section of mRNA molecule as shown below. Original sequence is given by this combination or arrangement of nitrogen bases, and then the mutant one give us that, and then name the type of mutation that is occurred. Of course, you can see this is called gene. It is called gene mutation, and then give reason for your answer is because there is a change in the sequence of nitrogenous bases from CC to CAG. We are seeing that if you see, we have got a change, a mutation that occurred in this party, which you can see that it changed from CC to C to CUG. So it is because there is a change in the sequence of nitrogenous bases. Change in the nitrogenous bases, which we can see that they changed from this one that you see, it changed from CCG, and it changed to, and then it changed to CUG, as you can see in the table above there. That's the change that we have there. There is a change in the sequence of nitrogenous bases. And then the next question, it says, the table below shows mRNA codons and the amino acids that they code for. Of course, that is what we are given there, the mRNA codons and the amino acids. And then the question is saying, state the number of different amino acids coded for by the original sequence of mRNA molecule given above. So the number, state the number of different amino acids, that are coded for by the original sequence of mRNA molecule given above. So now you can see that how many amino acids are we having from this. If we can see, the mutation is occurred on a section of mRNA molecule as shown. We've got 1, 2, 3, 4, 5. But then we need to look at the amino acids that are represented. AUG, it represents methionine. GAA, it represents glutamic acid. I can say 1, 2. We have got AUA, it represents isoleucine. We have got CCG, the original. 1, 2, 3. So we are saying the original, this one is the one that muted. Mutation, which we are saying, so it also has CCA, it represents proline. And then we have got GGA, it represents glycerin. So we are saying the number of amino acids that are on the number of different amino acids coded for is equals to 5. Give the anticodon on the tRNA molecule that carries the amino acid the anticodon. So we are given here that the mRNA, so we are looking at this one. So that is what we are looking at. So give the anticodon, we are given A, UA. Obviously, this is the codon. So give the anticodon that carries the amino acid, that one. So that is supposed to be, we are saying we are saying this one can be A. Obviously, it codes the anticodon is going to be U. U is going to be A, and A is going to be U. These are the anticodon class. The codon, anticodons. So what happens is that, of course, you remember to say this is mRNA and this is close to tRNA. So as much as you should see this, you should always just give the corresponding anticodons according to the complementary pairing of bases. Therefore, this can give you that it's going to be, this is going to be U, A, U, A, U, U, A, U, which you can see there. And then we are saying use the information in the table to describe the effect of the mutation that is occurred. So when you are looking at that, in describing the effect of the mutation that occurred, you always have to understand that you look at this to say, where did the mutation occur? The mutation occurred. If you look on the table here, class, the mutation is occurred. This is where the mutation occurred. That's where we have the mutation occurring. So mutation, where it occurs, it has got CCG changed to CUG. That is where we have the mutation. CCG, it changed to U, it changed to C, it changed to CUG. That's what the change is. So now, what is the effect of the mutation? CCG, it codes for proline, and then CUG, this one, it codes for leucine. That is the effect of the mutation. So now, what we need to understand is that the codon changed. It changed. The codon changed to, we have got the codon, it changed. We are saying this one, the codon that we have, our codon, which we are saying CCG, changed to CUG. The codon changed, the codon CCU, it changed to CUG, which I have just shown you here. This is the change that we are talking about. It changed there to there. So now CCU changed to CUG. Also, the anticodon sequence from tRNA changed as well. It changed. So in that case, we have got we said that we have proline. Proline is the one that we have, and then it gave rise to leucine. So proline, we have got proline amino acid, proline amino acid was replaced. It was replaced by leucine. It was replaced by leucine, and of course, this resulted in a different protein being formed, resulting in a different protein being formed. So obviously, because leucine has been proline has been replaced, so it's a new protein. It's a new protein structure altogether that is being formed. So if it didn't change, we would say that there is no effect on the on the uh, mutation. Mutation resulted from proline changing to leucine. So there is an effect in the type of protein that is being formed in that reference. There is an effect on the type of protein being formed. So we can go to question. This is question C. We go to the next question. The number of chromosomes in a somatic cell of organism differs from species to species. Yes. So the graph shows the number of chromosomes in each somatic cell of 3 different species. Number of chromosomes in 3 different species. So this is a bar graph anyway. So number of chromosomes is given, and then the species, we have got 1, the giraffe, 2, the mouse, and 3, the pineapple plant. So how many chromosomes will be present in mouse cells during telophase? How many chromosomes will be present in mouse cells during telophase 2 of meiosis? I'm sure you remember the process of meiosis. So during telophase 2 of meiosis, how many chromosomes will be present in the mouse cell? Mouse cell, it has got 40. We have got 40 originally. We have 40 there. So what happens is that during telophase 2 of meiosis, it means they should be halved. It is 40 divided by 2, which is equals to 20. It is 40 divided by 2, which is equals to 20. A leaf cell of pineapple, a leaf cell of pineapple, a leaf cell of pineapple. This is the number of leaf cell. It is 50. So in the what happens is that the leaf cell of a pineapple has the total number of chromosomes. So it is equals to total number of chromosomes that are present in the pineapple. So which is equals to 50. They're saying this equals to 50 because it's equals to total number that are in the pineapple equals to total number. So therefore it remains there. And then explain why the sperm cell of a giraffe has 15 chromosomes. The sperm cell of a giraffe has 15 chromosomes. A giraffe has got 30. I think you already know now. A giraffe has got 30. So this means that the giraffe is actually this is because it is a gamete. It is a gamete. So uh, which is formed by meiosis. So uh, and hence it must be a haploid. It must be a haploid, which means that a haploid that is 30 divided by 2, which is equal to 15, in order to prevent the doubling effect, in order to prevent the doubling effect, a gamete which is formed by meiosis must always be haploid. So haploid, it is always saying the total that we have divided by 2, which is 30 divided by 2, is equals to 15, that we are saying that it has there. So in order to prevent or avoid doubling effect, and then name the phase of meiosis where the halving of chromosomes number begins. The halving of the number of chromosomes, it actually begins at anaphase. It begins at anaphase. Describe the events in phase named in the above in anaphase. So what happens there is that during anaphase, spindle fibers they contract. The spindle fibers they do contracting. The spindle fibers contracting, chromosome pairs separate, chromosome pairs they actually separate, and of course move to opposite poles. They move to opposite poles. That is one thing that you should understand. You are just describing the process or the events that occurs during anaphase. And then we go to the next question. Information about blood groups in certain population. So basically we have got 4 blood groups based on A, B, O blood typing. And then one thing again is that blood groups is controlled by 3 alleles. You must know that how many people have the genotype, or how many people have the genotype, or number of people who have the genotype, or is actually the number of people which is given there, that is equals to 954,000 people have got blood group O. And then the next question is saying the population size is 1,800,000 people, and then calculate the value of X, show all your working, 2.4.2, calculate X. X is this one. So we are saying the number of people which are there. In order for us to calculate X, we can say that this can be calculated by saying the total number of people there. We have got 34%. We have 34%, and then which is 34% of 1.8 million. That is 34 over 100 multiplied by 1.8 million people over 1, which is equals to, when we calculate that, it can give us 612,612,000 612,000 people are the ones which are blood group, which is equals to X, because 34% of the total population, which is what? 1.8 million people. Describe how child inherits the blood group inherited by 3% of this population. 3% is AB, that is 2.4.3. How a child inherits blood group, that is blood group. That is blood group AB. So now, what happens is that a child inherits the blood group A is inherited from a parent one, and the other allele is inherited from each of the parent, and then the child would have a genotype of blood group AB. The child inherits allele A from one parent and allele B from the other. From the other parent. That results in a child being blood group AB. So what happens is that allele, we are seeing in this regard that the child is inheriting allele A from one parent and allele B from the other parent. When combined together, allele A and allele B, when they are combined together, we are going to have allele A and allele B, which is equals to blood group 1, which is equals to blood group AB that you see there. So it is parents who are contributing each that allele there. And then we are given this represent profiles of 3 children and their parents. Only 2 of the children are their biological children and 1 is adopted. This is one question which has to do with DNA profiling. Identify 2 biological children from this, the mother and the father. So the mother and the father. So identify 2 biological children from this. So the biological children must have identify the biological children who are, in this case, we can say, let's look at Ella. Ella, we have got Ella, she has got this, she has got that. Ella, she has got this, which is that, she has that, she has that. So Ayla can be a child of this family. And then if we look at Priya, she has this from the father, which is of course somehow less related from the father. Uh, and of course, if you see again, this one and there and there, so she cannot have only that one from one parent. So this one we might put aside. We have this one is from there. We have this one is from there. We have this one and this one is from there. And then this one is from there. So we can say that the children who are here, it can be Ayla and Leo. They can be the parents, the children from this. Explain your answer in this question. What happens is that question 2.4, 2.5.2, what happens here is that the DNA bands from Ayla and Leo match with the DNA bands of the parents. DNA bands from Ayla and Leo match to those of the parents. Match to those of the parents. That is why we can say that they are the what? They are the children. And then state the other uses of DNA profiling are 2.5.3 uses of DNA profiling. We have got the uses of this. It can be used for many. DNA profiling can be used for matching tissues for organ transplanting. Organ transplanting. DNA profiling can be used for, it can be used for identification of genetic disorder, identification of genetic disorders can be identified through DNA profiling. It can also be used in identification of suspects or solving criminal matters, resolving criminal cases. DNA profiling can be used in resolving criminal cases. It can also be used in tracing persons who are missing. Tracing missing persons. We can give men even identifying decomposing bodies. We can also use that. And then we have got question 6. Brown enamel of the teeth is a sex-linked trait. A dominant allele on the X chromosome causes brown teeth in humans. And then the question is saying, explain why more males than females have white teeth. 2.6.1 is sex-linked. These are sex-linked conditions. So what we should understand is that males have only one X chromosome. They have only one X chromosome, and of course, Y chromosome does not carry the disease-causing allele, and their Y chromosome do not carry the disease-causing allele. So now, they only therefore have to inherit, they only have to inherit one recessive they only have to inherit one recessive allele to have white teeth. They only have to have one recessive allele for white teeth, unlike for women, unlike for female, who have two X chromosomes. We have two X chromosomes. They have two X chromosomes, which may need them to have two recessive alleles for them to have the condition, for them to have white teeth. So many basically they have got one X chromosome, which that X, if it is affected, it is affected. Unlike for women or a female have got two X, which means that for them to be affected, both X's must have the condition, which is of course a lower case or a lower, which has got a lower possibility for that. And then a man with brown teeth married a woman with white teeth, use a genetic cross to show the possible phenotype ratios of their children. So we can say P1, phenotype. So P1 phenotype is a man with brown teeth. So we are saying male, brown teeth. He marries a woman with white teeth. Marries a woman, female, white teeth. So we need to have our genotype. This also class, it has been worked severally on metric hub, the possibilities of these. So we have got brown teeth. We say that the condition, we say that this condition is caused by, so brown teeth is sex-linked and is dominant on the, is sex-linked, a dominant allele on the X chromosome causes the condition. So brown teeth is a sex-linked and a dominant allele on the X chromosome causes brown teeth in male, in humans. So this is equals to say XY, and then he has brown teeth, and then he marries a female who is having white teeth. So a white teeth, it means they are X, but small b, which is going to say meiosis. And then we have got our gametes. Our gametes, we have got XB, and then we have got Y. We have got XB, and then we have got Y, and then it's crossed with X. It's crossed with XB, because remember this is 2XB. So if we can cross this, it can therefore, we have got this X and that X. We have got XB, this one, and that XB, and then we have got this Y and this one. We have got an XBY. So if we give this one, we have got one carrier female as to one white tick. One white tick son. That is what you are going to get, which is to say one carrier female and one white teeth son is what we can get in that regard. Which of course I'm saying, if you look at this, this we can also give it to say that this we can give it to say, remember we said this can also, this is equals to B. We said B represents brown teeth equals to one female or a daughter, one daughter brown teeth, one daughter brown teeth is to one son white teeth. That is 50%. 50% brown is to 50% white. 50% brown is to 50% white. That's what you do there, and then you get all your marks. You get all your marks. It is given. Let's just have the last question very fast. Read the extract. When a child is born, the umbilical cord is cut and stem cells can be obtained from it. Many people think that the stem cells for treating human conditions should be obtained from umbilical cords rather than from human embryos. Recent stem cells have also been obtained from bone marrow. These stem cells are used to treat conditions such as heart diseases and spinal injuries. So the question is saying, name 3 sources of stem cells mentioned in the extract. The sources of stem cells that are mentioned in this extract We have got the umbilical cord. We have got the umbilical cord. We have got the bone marrow, human embryos. We also have the bone marrow, which is equals to say 3.1.1 is umbilical cord. Umbilical cord. We have bone marrow. And then we have got the human embryos. There are the three sources that are there. Explain why the characteristics of stem cells make them useful for treating some disorders. This is because 3.12, this is because stem cells are undifferentiated and have potential to develop into any type of cells. Stem cells they are undifferentiated and have great potential to develop into any type of cells. That's why you see that into any type of cells, replacing affected ones. So stem cells, they are undifferentiated and great potential to develop into any type of cells, replacing the affected cells. And then name one condition in the extract that can be treated with the stem cells. We have got conditions to treat. We are saying heart diseases, can also treat spinal diseases. So it can treat heart diseases. It can also treat spinal injuries. It can treat such conditions, spinal injuries or heart diseases. And then read the extract. Flamingo and velvet are two species of monkeys that occupy the same habitat. Researchers recently discovered that a population of flamingo monkeys were able to interbreed with the velvet monkeys to produce offspring. These offspring were infertile. They were infertile. That is a question for life science students. And then the question is saying, define a population. 3.21 population is a group of organisms. Population is a group of organisms of the same species. That of course occupies the same habitat. That occupies the same habitat and live together, able to interbreed and are able to interbreed. And they are able to interbreed. That is a population. That is a population group of organisms of the same species that occupy the same habitat and are able to interbreed, we say that they are a population. And then give one reason why velvet monkeys are considered to be two different species. 3.2.2, they are they are considered two different species because the offspring that they produced was not able was infertile. They produced an infertile offspring. They produced an infertile offspring. It's called hybrid sterility. In other terms, they produced an infertile offspring. So therefore, if they are producing an infertile offspring, that can be defined as what? As being different population species. These three mechanisms of reproductive isolation that are not mentioned above. I said that speciation, you always need to be quite understanding and you need to prepare yourself for it. We have got one, we can say reproductive isolation. It can be caused by breeding in different times of the year. Different times of the year. Breeding different times of the year can be the reason. We also have adaptation to different pollination. Adaptation to different pollination, to different pollinators, agents of pollination are different. We can also have caused by species-specific courtship behavior. Different species courtship behavior. That is to say that the courtship behavior they exhibit can be different. And then I may lastly say that it can be because of gametic isolation. Where the gametes cannot be able to fertilize each other. That can also be a cause of, it can also cause reproductive isolation. And then scientists find evidence of human evolution by comparing humans to other hominids. The upper limbs of humans and African apes should show similar characteristics, whereas there are differences between detention of the two. Why do scientists look for similarities between humans and apes? Why do they look for similarities between humans and apes? That is basically to show a possible common ancestor or to identify trends in evolution. To identify trends in evolution. To identify trends in evolution, can we trace that in order to identify trends in evolution? It can be the reason for that. To identify trends in evolution and also explain the importance of positioning of the thumb for humans and apes. Actually here, you need to know that both the the humans and apes, they have got opposable thumb for a precision grip. Both they do have an opposable thumb for precision, or we can say a tight grip. A tight grip must be obtained by the opposable thumb. So it gives a tight grip, which we call a precision grip. That is that state that one difference between the teeth of humans and African apes, the difference between the teeth of these humans, actually, the humans have got smaller teeth. Humans, humans have smaller. Humans, they do have smaller teeth or canines, whereas African apes have large teeth. African apes, they have large teeth or canines. Human teeth, human teeth, they have no gaps. They have no gaps between them. Whereas African apes, teeth have got gaps. I guess you remember to say that is called, which we say that is called the diastema between teeth. I remember this term is the scientific term. They have got the diastema. So those are the differences that we can give with reference to question 3.3 between the human teeth and African ape's teeth. And then human evolution, we are given this evolutionary tree that is shown. I'm always saying that it comes in several ways, or it is presented in different formats. So what type of graph is this one? What type of graph is it? You shall see that quite a lot of times it is presented in different formats. So I was saying that you should always know that whenever it comes, be able to understand the different ways that it is being asked. You should know the different ways through which it is asked. And then what is the name of this tree, of this diagram? We call it a phylogenetic tree. It is called a phylogenetic tree. It's called a phylogenetic tree. Others, they call it a cladogram. Others, they call it a cladogram. And then how many genera are represented in the diagram? How many genera are represented in the diagram? We have got we have got the Homo, which is Homo erectus, what, and then we have got the Australopithecus africanus, and what. So we basically have these. We have got two. We basically have two of them, and then name the species represented by X on the diagram. X, we have got Australopithecus africanus. It actually gave rise to Homo ratofensis, and then this gave rise to, you must understand that it gave rise to the Homo habilis. The Homo habilis resulted in Homo erectus and the Homo naledi. So Homo naledi is a species which developed from Homo habilis. The Homo habilis. The Homo habilis is X. The Homo habilis is X. And then name the species that share a common ancestor with the Homo erectus. Homo erectus. Homo erectus is this one. So it is actually existing at the same time. We are saying this one and this one are existing at the same time, which is to say sometimes they say give the common ancestor of Homo erectus and Homo naledi. So the common ancestor for this is the closest common ancestor is the Homo habilis, which used the stone tools. The stone tools were done by Homo habilis. So this B, it is actually representing, or it is the Homo naledi that you can see there. Homo naledi. Homo naledi is what you can see there. Which species of the genus Homo is only one in existence today? Which species of the genus Homo is the only one in existence today? The only one in existence today, we are saying we have got the Homo sapiens. The Homo sapiens, the modern man, is the one that is in existence of now, which is this one is the Homo sapiens. The modern man is the Homo sapien. Name two forms of evidence that would have been used to support the information in the graph. So, sources of evidence for evolution. Sources of evidence for evolution. We have got these can be, we have got the sources of evolution, we have got cultural evidence. We have cultural evidence. We also have genetic evidence. We can also use fossil evidence. They can be used to define evolution or to explain evolution. And then the average cranial capacity of Homo sapien is 1,500 cubic centimeters compared to 520 in Australopithecus. Explain the significance in difference in cranium capacity. Cranium capacity, what you need to understand is that a large cranium capacity in Homo sapien indicates large brain leading to greater intelligence. So, a large cranium capacity, in Homo sapiens In Homo sapiens, it indicates large brains. Indicates large brains, which of course resulting which result in greater intelligence. Which results in greater intelligence. That is the difference in cranium capacity 1,500 versus the 520. So the smaller the cranium, the smaller the brain. Also the less thinking capacity. That speech is 3.47. Explain how a fossil of Australopithecus africanus, species X and Homo erectus are used to support out of Africa hypothesis. Out of Africa hypothesis, what it does is that it actually identifies that fossils of Australopithecus are the oldest fossils. We can say that the fossils of Australopithecus were found in Africa only, and of course the fossils of species X were found elsewhere. Fossil of Australopithecus the fossils of this species were found in Africa only. In Africa only. And other fossils of the Homo habilis. Homo habilis. The fossils of the Homo habilis were found in Africa only. And then, of course, the oldest fossils of Homo erectus were found in Africa. What we are saying is that, how do we say that the first humans were found in Africa? It is also explained by the fact that oldest fossils were also the oldest fossils of Homo erectus. The Homo erectus were found in Africa. They were found in Africa. And then, of course, indicating that modern humans originated in Africa and migrated out of Africa. All these evidences indicates that modern humans originated in Africa. They originated in Africa and migrated out of Africa. Which we call out of Africa hypothesis. And then next question. It is modern day whales and aquatic mammals spending their entire lives in ocean. they are thought to have evolved four-legged ancestors as represented there. So we are given these ones: quadrupedal carnivore, the flipper, flat feet, large flipper, and what, and the present day non-functioning pelvis, large flippers in front. So which ancestor of whales most likely lived both in water and on land? Which most likely lived in both water and land, which we can see that it has got flipper, large feet, flipper like large feet and tail of swimming. So as you can see, quadruped carnivore, it could not live in water, but we are seeing flipper for swimming, and also the tail, the flipper like feet and the tail for swimming. So it is the ampulators. It is the ampulators because it has got features of both. Give one reason for that. It had flipper like leg, like large feet, and also a tail, which are for both. It has features to survive in both water, and land. In both water and land. As you can see, we are saying that large flipper like feet and a tail. So that is why we can say that it survives both environments. And then explain why abulitas and the Duradon may be considered as transitional species. They are transitional species is because these are transitional species. They actually this is because they share characteristics of the ancestor. These are transitional species, this one and this one. They have got the features of the ancestor, which shows that they are actually transitioning or changing. They share characteristics, they have characteristics, or they have intermediate characteristics. They have intermediate characteristics of the ancestor. And the present day species. The ancestor which you see that the ancestor which we say that this is the ancestor, this one. And the present day, in the present, the present day. So they have intermediate characteristics of the ancestor and the present day species. Present day species, which we can also see that it is the balana. If something is transitional, it has the characteristics of the previous and the current or of the present day. That is it. Explain according to Lamarck why modern day whales do not have legs. Is the theory of use and disuse according to this guy. According to this guy, the ancestor species 3.57, 3.5.4. We say that the ancestral species of the whales, ancestral species of the whales all had legs. Species of the whales all had legs. They had legs as more time was spent in the water. More time was spent in the water. Of course, searching for food. The legs were used less and disappears. And they disappeared. And they disappeared. So the acquired characteristics was passed on to the next generation. Acquired characteristic was passed on to the next generation. That's what happens then. At first, that is why we said it had large flipper-like feet and also a tail, which of course these are showing that at first it was living on land and then it started searching for food in water, making the legs less useful, and then the legs, they disappeared, resulting in them having the water characteristics, which they were now passing on to the next generation, resulting in the entire population of whales not having legs but living in what? Living in water. That's what happened. And then we have got patients infected with HIV drugs. HIV/AIDS are treated with ARV. When they miss their treatment, it can increase chances or probability of the virus. Scientists conducted an investigation to determine the effect of the number of missed treatments on the probability of HIV virus developing resistance to the drug. So results are shown ends below. This also you can check on Metric App again. Check on Metric Hub. It has been explained and answered also at the evolution in present times. Evolution in present times, it was well explained. So what we are looking at, results are shown, days, number of missed treatments, probability of the virus developing resistance to ARV. So as the days increases, the percentage probability increases also. So state the following. State the actually an investigation was determined to determine the effect of mistreatment on probability. That is where the dependent and independent variable is coming from. So our answer of dependent and independent comes from the investigation. So now we are saying, if you look at this, you should see that the dependent on the effect of number of mistreatments on probability of HIV virus developing resistance. So the dependent HIV, we are saying the dependent is probability of developing resistance to ARV is the what is the dependent is the dependent. This is the probability of developing resistance probability of developing resistance to ARV drugs. And then the independent variable is number of missed treatments. Number of missed treatments. It is the independent. Number of missed treatments. Number of missed treatments is the independent variable. And then based on the results, state one precaution for patient receiving ARV treatment actually is to actually must not miss. Precaution is not to miss the treatment. Actually, treatment must not to miss treatment. Treatment must not be missed. Treatment must not be missed. And then state a conclusion for this investigation. The conclusion, as you can see, we are saying as the days increase is going down, the percentage probability also increases. So what happens is that the more the days of missed treatment, the greater the probability of virus developing resistance. That's the conclusion. The more the days of missed treatment, the more the days of missed treatment, the greater the probability which we see of HIV developing resistance. Of HIV developing resistance, which of course developing resistance to ARV drugs. This is shown here. 2 days probability is 0, 7 days 20, 21 days 40, 37 days 60. So the more the days, the greater the probability of HIV developing resistance to what? To ARV drugs. That's what we can see as a conclusion there. And then describe the evolution of resistance to ARV medication in HIV virus. I'm saying this one again, it's also on Metric Hub, guys. It's also on Metric It is also given on Metric Hub. Look on evolution in present times, the treatment or the control of uh, antibiotics, resistance of bacteria to antibiotics. So in this case, you should understand that there is a great variation in resistance of HIV virus to ARV drugs. So some viruses are resistant to the drugs and others are basically not resistant. And those that are not resistant do not survive. The the ones which are not resistant to the ARV, they don't survive when treatment is missed. So the resistant viruses survive and reproduce, passing on the resistant gene or characteristic to their population or to their young ones, resulting in an increase or population of resistant uh, viruses to ARVs. That's what you should say. So I'm saying it is also on Metric Hub class. Metric Hub evolution in present times. What I need you to look at is that please go to the playlist. We have got quite a lot of content on life science, which if ever some of you or for those who are following, you will never regret because all those videos that are presented here, they are quite comprehensive and so uh, preparatory. They are so loaded to the extent that you won't miss up your exams. You actually excel. So thank you so much for following and let's keep connected until we meet again. I have a great and nice time. Preparatory exams are coming term 3 and also we also have those who are writing the SBA. I'm going to represent, I'm going to actually work them out with you. Please don't finish the videos. Sign in now by signing to join and then follow the videos to the end. You won't regret this. Thank you so much and have a great term. Have a great term. Bye.